Assalamu alaikum. I am Dr. Mouly Tabassum. Today we will discuss regarding the fundamentals of genetics. So, we will first discuss about what is genetics. It is the science of genes, heredity and variation in living organisms. Now, what is genetic material? Nucleic acid which contain genetic information are known as genetic material. So, genetic materials are the materials that contain the genetic information. For example, in human, it's DNA. So, what is nucleic acid? Nucleic acids are polymers of nucleotides. Nucleic acids are polymers of nucleotides. They are held by a 3' to 5' phosphodiester bond. So, what is nucleotide? Nucleotides are the structural unit of nucleic acid, composed of nitrogenous base, pentose sugar and phosphate group. So, nucleotide is the combination of 3 different units containing a nitrogenous base, a pentose sugar and a phosphate group. For example, AMP, adenosine monophosphate. It contains adenosine as a nitrogenous base, a pentose sugar and a phosphate group. ADP, that contains adenine, a pentose sugar and 2 phosphate groups. And ATP contains 1 adenine, 1 pentose sugar and 3 phosphate groups. In nucleotides, nitrogen bases are attached with the first carbon and the phosphate with the 5th carbon of the ribose sugar. So, what are the functions of the nucleotides? These are the structural unit of nucleic acid. Acts as second messenger, energy currency of cell and acts as coenzyme. It also acts as carrier of active molecule in synthetic pathways and also allosteric effector to regulate several enzyme activities. Now, what is nucleoside? Nucleoside. Nucleosides are the base and pentose sugar. So, in case of nucleotide, there was a phosphate group and in case of nucleoside, there is no phosphate group. Only the base and the pentose sugar is form the nucleoside. For example, adenosine which contains 1 adenine and 1 pentose sugar and cytidine which contains cytosine, a nitrogenous base and a pentose sugar. So, what are the functions of the nucleosides? These are the precursors of nucleotides. Are act as signaling molecules. Altered nitrogen bases in nucleosides have a role in regulating genetic information. Nucleoside analogues are used in the treatment of malignancies and viral diseases. Now, we will discuss regarding the different components of nucleotides. First, nitrogenous base. What are the nitrogenous bases? The bases are aromatic, heterocyclic compounds. The bases are aromatic, heterocyclic compounds. There are mainly two types of bases, purine and pyrimidine. There is 2 purine base and 3 pyrimidine bases. Major purine bases are adenine and guanine and minor bases consist of xanthine, hypoxanthine and uric acid. In case of pyrimidine bases, major pyrimidine bases are cytosine, thymine and uracil. Pseudouridine and dihydrouridine are minor pyrimidine bases. Base atoms are numbered as 1, 2, 3, 4. In case of purine, there is 9 atoms which are numbered anti-clockwise, and in case of pyrimidine, there is 6 atoms and they are numbered as clockwise. In case of sugar atoms, they are numbered as 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. Now, how the purine nucleotides are synthesized. The raw materials for synthesis of purine bases are aspartate, glycine, glutamine, carbon dioxide, N10-formyl tetrahydrofolate, and ribose 5-phosphate. They are mainly synthesized in the cytoplasm of the hepatocytes. First of all, there is synthesis of PRPP, phosphoribosyl pyrophosphate, and activated pentose. So, ribose 5-phosphate with the help of ATP forms the PRPP or phosphoribosyl pyrophosphate. This phosphoribosyl pyrophosphate then converted to 5' phosphoribosylamine. Here glutamine is converted to glutamate. Now, synthesis of INP or inosine monophosphate, the parent purine nucleotide. 5' phosphoribosylamine with 9 different steps converted into inosine monophosphate. Conversion of inosine monophosphate to AMP and GMP is the last final pathway. Inosine monophosphate with the help of GTP and aspartic acid forms AMP or adenosine monophosphate, and inosine monophosphate with the help of ATP and glutamine forms GMP. Synthesis of deoxyribonucleotides are formed from ribonucleotide diphosphate. These ribonucleotide diphosphates with the help of ribonucleotide reductase forms deoxyribonucleoside diphosphate such as DADP, DGDP, DCDP, etc. Now, how the catabolism of purine nucleotide occurs. AMP with release of phosphate forms adenosine. This adenosine with release of ribose and ammonia forms hypoxanthine, and with the help of xanthine oxidase they are converted to xanthine. Similarly, GMP with the help of with release of phosphate forms guanosine, and from guanosine ribose and ammonia comes out and forms xanthine. This xanthine with the help of xanthine oxidase forms uric acid. So, the end product of purine catabolism in human is uric acid. Purine rings are not cleaved in human. There is a condition known as gout, in which it is a increase in uric acid accumulation occurs. So, gout is a condition or metabolic disorder of purine catabolism and characterized by hyperuricemia. Salvage pathway of purine. Purines that result from the normal turnover of cellular nucleic acids or that obtained from diet and not degraded can be reconverted into nucleoside triphosphates and used by the body. This is referred as the salvage pathway for purine. So, purine nucleotides. Normal purine are not catabolized fully in our body, and it is used again in our body. Two enzymes are responsible for the salvage pathway. These are APRT and HGPRT. APRT or adenine phosphoribosyl transferase and HGPRT or hypoxanthine guanine phosphoribosyl transferase. Now, synthesis of pyrimidine nucleotide. Raw material for pyrimidine nucleotides are, for the base, glutamine, carbon dioxide and aspartate, and ribose 5-phosphate. First, synthesis of carbamoyl phosphate occurs from carbon dioxide and glutamine. Then, synthesis of orotic acid from carbamoyl phosphate and aspartate, and formation of pyrimidine nucleotide OMP, orotidine monophosphate, the parent compound of pyrimidine synthesis, from orotic acid and PRPP. Synthesis of UMP, CMP and TMP occurs. Catabolism of pyrimidine nucleus is much simpler. Pyrimidine nucleus with release of the phosphate group converted to pyrimidine nucleoside. And after the release of the ribose sugar, the nucleoside is converted to pyrimidine base. This base is further converted into beta-alanine, beta-amino, isobutyrate and ammonia and carbon dioxide. Pyrimidine nucleoside can be salvaged by nucleoside kinase. Salvage of nitrogenous base. Free purine and pyrimidine base is produced by catabolism of nucleic acid without being degraded and excreted as waste. They are usually reused by the body. This is called the salvage of nitrogenous bases. By this way, both the purine and pyrimidine are in the form of salvage pathway. So, this is enough for today, and these are the fundamentals of genetics. We discussed some of the key words of genetics today. Thank you.